Okay, good afternoon students. The last class we were discussing about two different topics. What is the menses cycle? Then also we dealt with gametogenesis. In gametogenesis we know a sperm and egg cell which is haploid will fuse to form a diploid zygote. Gametogenesis takes place in two different aspects. One is in the production of sperm cells called spermatogenesis. Then production of egg cell is called oogenesis. Okay. Now we are going to study another topic, that is fertilization and implantation, maintenance of pregnancy. Before that, I just recap what we have studied in female reproductive system. With that, the female reproductive system will get over. Now this is the ovary of a female, and we know a female child is born with 1 to 2 millions of primary oocyte, and before birth all the oogonia, primordial germ cells or egg mother cell, will get converted into primary follicles which is diploid in number, and after birth no more oogonia is added. That is what we study in this ovary. Now after the birth till puberty, the primary oocyte is undergoing an arrest. Nothing is being developed. That means cell is not undergoing division. By the time the follicle which is surrounding the primary oocyte will get converted into secondary, tertiary, and get matured into Graafian follicles. By the time the primary oocyte within this ovary will get converted into secondary oocyte, and at the time of ovulation, that is on 14th day, that is during the 14th day, one egg which is fully, that is secondary oocyte, which is fully matured, ruptures the Graafian follicle and enters into this tube called fallopian tube. Okay, so now this fallopian tube, if you take, it is having three different areas. A finger-like structure will be there, that is called fimbriae. Okay, and we will study all this when we study about the structure of female reproductive organ. Now this is the fimbriae, and the tube which is extending towards this is called the fallopian tube. Otherwise this tube is called oviduct. We call this oviduct because duct is a tube that carries a ova, so it is called oviduct. So fallopian tube is also called as oviduct. The fallopian tube, if you take, I told you three parts are there. A finger-like projection called fimbriae, and this area is called ampulla. Very important structure. In the ampulla only, fertilization will take place. After the egg liberates from the ovary, enters, and it remains in the ampulla for 24 hours. So in this ampulla, the fertilization will always take place. It's a public 1 mark question. Okay. So soon after the egg comes, it is on 14th day of ovulatory phase. It remains there for 24 hours, and that is what I have put it here. What is happening inside, I just put it out. Here the egg will be there. By the time the sperm cell has entered into the female reproductive tract, fuses with the egg cell, it becomes fertilized. That is what we are going to study in this fertilization process. We studied if the egg is not fertilized, it is undergoing menstrual cycle. That is what we studied in the previous classes. Now what we are studying is once the egg is getting fertilized, now what is going to happen in the tube, and ultimately that fertilized egg develop into another structure, and it is getting implantation, that means fixing into the wall of the uterus. How all this is going to happen? That is what we are going to study in this topic called fertilization and implantation. Fertilization, as I told you, egg and sperm cells are fusing together. So here in fertilization, you have got the haploid number of cells. That is the haploid number of the sperm cells fuses with haploid number of egg cell. As a result, you get diploid zygote. So these are the important one when you talk about the fertilization process. Clear? Now as you know, where the fertilization is taking place? Inside the area of the fallopian tube called ampulla. So there the fertilization is taking place. Okay. And the end part is called isthmus. This area is called isthmus, where it is connecting towards the uterus. Understand? So this is the uterus, where this uterus will be lined with this membrane. That is what we studied. This membrane is called endometrium. This membrane is called endometrium, where the egg comes and fixes on it. Every month, from this month, this ovary, next month, this another ovary. Okay, so it is coming and fixing, right? So it's an interesting topic what we are going to discuss now. Fertilization, how we are formed in the womb of the mother, how we develop, all this that we are going to study here. So now, so it is fertilization is haploid, sperm and egg fuses to form the diploid zygote. Where is the fertilization taking place? It is in the fallopian tube or it is in the ampulla? It is the ampulla of the fallopian tube. Understand? Right. So now the next one is the topic that we study here. It is capacitation. Very important. 2 mark question. Afterwards you can mark this one. See, capacitation is, you know, is active ability. So here we are talking about the capacitation of these sperm cells. The capacitation of the sperm cell. Now what is the capacitation of the sperm cell in fertilization? What is the capacitation of the sperm cells in fertilization? The sperm cell has to enter into this female reproductive tract, okay, and enters into this area and goes to the fallopian tube. By the time egg is here and the sperm cell fuses with this egg cell. Okay, so that is what capacitation of the sperm cells. That means a sperm cell has the capacity of entering into the female reproductive tract. Number 1, it can enter inside. Number 1. Second one, as soon as entered inside, it has got a capacity of fertilizing an ovum or egg cells. So that is what the sperm cell capacity is. It has the ability of entering into the female tract. It is having the capacity of fertilizing the egg cells. But if fertilization should take place, the egg and the sperm cells should meet simultaneously. So if it is ovulatory phase, if the female is undergoing reproduction, I mean coitus in the sense having a sexual intercourse, that time the sperm cell enters into that and it is getting fertilized. Only that 24 hours, only that one day. If the egg is somewhere else or egg is just coming from the ampulla and if it is meeting here, nothing is going to happen. Is it clear? So fertilization, that is what we studied, always in the ampulla where egg and sperm cell simultaneously meet each other. Clear? Right. So that is what capacitation is. Clear? Right. The next one is, now this process, capacitation process is the biochemical event. It is a biochemical event. We will talk what this biochemical event is. Okay? Right. So now, next one is another topic called agrosomal reaction. Now, you know where the agrosome is. The agrosome is presented on the tip of the sperm cells. If this is the sperm cells, all this tail, everything is there. The tip part is the agrosome, cup shaped, small cup like structures, agrosome. Agrosome is made up of an enzyme called proteolytic enzyme, or it is otherwise called as sperm lysine, or otherwise called as ion uronidase enzyme. We studied, right? Now, this enzyme is helpful for the sperm cell to enter inside. If you talk about the egg cell is having this membrane called zona pellucida, and this one is called corona radiata. Okay. Now, these two membranes are fixed by an acid called, we studied, an acid called ion uronic acid. That is what we studied. Okay. Right. So now these two layers are fixing together by the enzyme called ion uronic acid. Okay. Now, this sperm cell, when it enters into this egg, it has to penetrate into this membrane, and then only the nucleus is sent to the egg cells. Okay. Now, the enzyme which is there in the agrosome is called spermlysin. You remember ion uronidase enzyme. Ion uronidase enzyme. Now, this enzyme will remove the agrosome, the tip of the sperm cells. Okay, that is why it is called spermlysin. Lysin means killing, destroying. So here the tip part is destroyed by this enzyme called ion uronidase, so that the tip removed and send the nucleus inside. By the time these two membrane also made a kind of a remove or disintegrated. Okay, so that reaction is called agrosomal reaction. Agrosomal reaction means sperm in the agrosome as an enzyme called ion uronidase enzyme. With that, it has the ability of penetrating into the zona corona radiata and zona pellucida and sends the nucleus towards the egg cells. And this nucleus and egg nucleus fuses each other. That is what the fertilization process is. So this is the process called the zygote. Understand? So two things you remember. What is capacitation? You know what is agrosomal reaction. Okay. One more thing, that is called polyspermy. See, poly means more. Sperm cells means more sperm cells. Now, there are around 200 to 300 million sperm cells comes out during coitus. Coitus, that's what we study, 200 to 300 millions of sperm cells are liberated at the time of reproduction. Now, among the 60% should have normal shape, 40% should have vigorous movement. Okay, so now out of this 200 to 300 million, this sperm cell, around 50 million sperm cells will be fighting with this egg cell to enter inside. How many? 50 million sperm cells will start fighting with this to enter inside. Okay, fighting in the sense what? Agrosomal reaction. Removing and make a holes in the zona pellucida and enter inside. Okay, now this zona pellucida has a capacity of, as soon as the sperm cell, one sperm cell entered inside, the zona pellucida makes one more layer on it called, I will write it here, called fertilized membrane. Called fertilized membrane. Okay, listen, as soon as one sperm out of 50 million, one sperm has entered into this egg cell, as soon as it gives the nucleus, the zona pellucida makes an alter to form another covering called fertilized membrane, and this fertilized membrane will not allow another or some more sperm cell to enter inside. So polyspermy is prevented, is avoided by a membrane called fertilized membrane. Who altered this membrane? Zona pellucida. Sperm cell enter into the egg or penetrate into the egg membrane to enter the nucleus into it is called the zona pellucida. It's a primary egg membrane. Okay, so this membrane got disintegrated and the sperm cell will pierce into this zona pellucida and enters. Understand? So these are all the follicle cells, corona, radiata and the zona pellucida. Is that clear? So now we know what is fertilization, where the fertilization is happening, what is capacitation, what is agrosomal reaction, then we know what is polyspermy. Correct? Right. Now we will study how this, after fertilization, the cell division, what are the divisions taking place and how it is coming and implanted. Very important, listen. Now, as soon as the egg is getting fertilized, how it is fertilized? Egg cell and sperm cell meets simultaneously. As soon as it is getting fertilized, now the cell will undergo divisions. Okay? The first division is two identical cells produced. Both cells are similar, two identical. Now, in this two identical cell, we are calling these two cells are called the blastomere. The first cell division after zygote is called blastomere. This is what? Okay, in your book you will find one small box will be there. Afterwards you can see it. That is called monozygous. Monozygous twins always. Last is public 1 mark question. Monozygous means both will be similar in sex and appearance. Both same sex will be there. Both are male means both are male. Both are female means female. That is called monozygous twins. Monozygous twins are found from always one egg, one sperm. Because last year most of the students have written monozygous means two individual, so two eggs, two sperms. Entirely wrong. Mono means you remember single. Okay, one sperm, one egg fuses. When it is dividing into two identical cells, after this two together form four. But here what happens in monozygous? After division of this will produce another individual and that will produce another individual with the same egg one. Okay, so monozygous are always one sperm, one egg. Dizygous means two egg, two sperm. Outside 1 month, one ovary liberates one egg. Sometime both the ovary liberates egg. So this side one fuses and this side one fuses. So two sperm, two egg. For dizygous, monozygous, one egg, one sperm. Understand? Mono means only both are identical twins. Sex will be both same. Male means male, female means female. Here, one egg, one sperm. Dizygous, both will be different. One male, one female will be there. Okay, so that's called dizygous, non-identical twins. So here, one egg comes here. At the same time, one more egg was also liberated. So two fallopian tube, two ovaries. Here also will get fertilized. Here also will get fertilized. Is that clear? Right. So now listen. As soon as the egg is getting fertilized, it is forming a cell stage called blastomere. Okay. After that, it forms around 4 to 8 cells, and then it forms around 16 cells. So all these together, here it is called the morula stage. Zygote, blastomere, and morula. Okay. Now everything is happening inside the tube. Now listen. This tube is little wider like this. See, I just put it here. This is wider, ampulla. And you see this tube, it is quite narrow. Okay, it is fixed, not opened properly. So, this is a problem in some cases. Now, this tube should open enough. Then only the egg can move and come here. Is that clear? Fertilization takes place here. Cell division, everything is taking place. After all the division, the egg should come and fix on the wall of the uterus. Okay. So now here, this is little wider. I can even put it like this. Like this is wider. So egg can easily stay there, remain there. That is the reason egg is remaining there for 24 hours. And after that, the egg is moving here. Okay. As soon as the egg comes here, how the egg will come? Because of an hormone called progesterone hormone. Progesterone. The progesterone hormone will open this fallopian tube, then the egg is moving towards the uterus. Okay, so this hormone is very important for opening of the fallopian tube. Understand? By the time the cell attains 16 stage of cell called morula. 2 cell divides into 4, 4, 8, 16 and more cells. Okay. Then it will come and fix in the wall of the endometrium. That is wall of the uterus called endometrium. When it comes to the uterus, it is in the stage called blastocyst. Blastocyst. So it is zygote, it is blastomere, it is morula, and finally it is called blastocyst. Understand? Right. So now when it comes to the blastocyst, it forms outer layer. This layer is very single layer. Single as well as flattened cells, or it is flattened layer. Outer part, flattened. So this outer part is called trophoblast. This is entirely it is called blastocyst. Outer part is called trophoblast. inner you will find some 20 to 30 rounded cell that is called inner mass cell or otherwise called as embryo. Okay, so once again I just tell you, the fertilization will take place in the ampulla where egg and sperm cell meets each other. Then after that it is undergoing a division called blastomere. Then you will have 4 to 8, then 16 or more called morula. Now by the time progesterone hormone opens the fallopian tube and the egg comes and settles down in the uterus or endometrium. By the time it is called blastocyst, and outer part blastocyst is made up of trophoblast, flattened single layer cell. Inner you will have some 20 to 30 inner cell called embryo. Understand? So this is how the cell is dividing and undergoing fertilization process. Clear? Right. Now here, suppose if this fallopian tube is not opened up by this hormone called progesterone. Okay, if it is not open, it has to open. Suppose if it is not opened up, 95% of the pregnancy will take place outside the uterus. That means the child development itself entirely is happening here in the ampulla. Actually, child development should take place inside the uterus. Only division. Within 4 to 5 days, the entire process is completed within 4 to 5 days. This egg should come and settle down here. Where? Into this endometrium. If it is remaining there till 9 months, that means 280 days. That is 40 weeks of pregnancy. Pregnancy lasts for 40 weeks, 280 days, 9 months around. Okay? So now this entire pregnancy takes place here, development takes place here. 95% it happens outside the uterus. Okay? So in that case, what will happen here? The child internal bleeding will take place and the fallopian tube will rupture and it will lead to death of the fetus and the mother. Clear? Because pregnancy should take place in the uterus. But what happened here? The whole entire 280 days will be taking place here means it cannot withstand, so it bursts and ultimately the mother and the child will die, and that is for 95% cases. This is called ectopic pregnancy. Now what is ectopic pregnancy? Is a 2-mark question. Okay, it is called ectopic pregnancy. 95% it takes place outside the uterus. Okay, so this is what fertilization and implantations are. Okay, now what we are going to study is maintenance of pregnancy. Maintenance of pregnancy means now the ovum, fertilized ovum is in the uterus. Okay, so now what are all the things that is going to develop in the baby, in the fetus? That is what we are going to study. Is that clear? Right. The next topic we will concentrate on the maintenance of pregnancy. Okay. See, the last topic that we discussed, fertilization and implantation, we studied zygote, blastomere, morula, blastocyst. Blastocyst will have 2 types of cells. Outer part is called trophoblast. Inner you will have 20 to 30 rounded cell called inner mass of cells. Now we will study now about this inner mass of cells, that is called the embryo. So inner mass of cell will form an embryo. Okay. Now embryo will consist of 2 things. In embryo there are 2 things are there. One is called epiblast. Epiblast means, see, this group of cell forms an outer layer. Outer layer is formed. That means inner organs are developed. Okay, the child is developing from this group of cells. Okay, when the child is developing from the group of cells, the first development is 3 layers will be formed around. Okay, so together the 3 layers are called primary germ layers. Okay, so as the child is formed in the womb of the mother, when it is developing, the inner mass of cell, that is 20 to 30 rounded cell, will develop 3 type of layers. Each layer will give a particular rise to the organs. Okay? That is the outer part. So this is called epiblast. Epiblast forms outer structure, so it is called as ectoderms. Ecto means outside. Derm is cell layer, skin. The second one in this embryo is hypoblast. Hypoblast forms the inner layer. This is the, otherwise it is called endoderms. Okay? Epi means outside, hypo means inner layer. Okay? In between that, you will find, in middle you will find one layer. This is called mesoderm. Mesoderms. Okay? So entirely the inner mass of the cell is divided into 3 layers. Okay? Epiblast, ectoderm, outer layer, hypoblast, inner layer called endoderm, and in between you will have mesoderms. Okay? Now each layer is giving rise to some particular organ, which is there on page number 23, right side, second paragraph. Second paragraph. See, first what we studied, page number 22, inner mass of cell, together all this together is called blastula. Together this inner mass is called the blastula stage. The stage is called blastula. Okay. So now this blastula will differentiate into epiblast and hypoblast. When it is differentiated? Immediately after implantation. Once it is fixing in the wall of the uterus, it is differentiated into epiblast. Till that it is not differentiated. Okay. Now the hypoblast is embryonic endoderm inside, and the epiblast is the ectoderm. The remaining in between the epiblast and the endoderm is the mesoderm. Okay. Now all these things will lead to a structure called blastula, will lead into gastrulation or gastrula. Gastrula means this 3 layers forming. Okay, the 3 layer forming is called the gastrulation. After this gastrulation, every layer will give rise to organ, so it is called organogenesis. Organogenesis. See, first one is called the zygote, second one is called the blastomere, third one is called the morula, fourth one is called as blastocyst, inner is called blastula, then it forms gastrulation, or that is 3 layer formation. Then each layer will give rise to organ, so they are called organogenesis. Understand? Right. Now, see that transformation of blastocyst into gastrula. With the gastrula will have always 3 primary germ layers, and all these germ layers will move into the blastomere. That means it will enter and occupy their own areas. Ectoderm, mesoderm, endoderm, all this, and form giving rise to specific organ, tissue, organ system under the process called organogenesis. Understand? Right. Page number 23, right side, second paragraph. Primary layers giving rise to ectoderm. See, all the ectoderm will give rise to central nervous system, CNS. What is CNS? Brain, spinal cord. So all the brain, spinal cord are formed from the layer called ectoderm layers. Understand? Not only that, even some peripheral nervous system, the neuron outside the central nervous system are called peripheral nervous system. Okay. Then epidermis, outer layer of the skin, and mammillary glands. All these, that epidermis and derivatives as well as mammillary gland. All these are produced from the ectoderm cells. What are all ectoderm? Central nervous system, PNS, peripheral nervous system, and epidermis and mammillary gland. So, milk producing gland. All these glands are produced from the ectoderm cells. Okay, right. Second, mesoderm cells, that is connective tissue, cartilages, bones, muscles, urinogenital system. That means kidney, urethra, gonads means reproductive organ. And last one, endodermal cells, epidermis of epithelial cells of gastrointestinal inside the stomach and intestine. They are lined with epithelial cells as well as respiratory tract, liver, pancreas, thyroid and parathyroid. All these are the structures which is produced from different layers. Definitely they will ask you one mark in the public exam. They will give one particular organ and they will ask you to find out from which layer it has formed. Okay, so far what we are studying is, we are studying the inner mass of the cell that is giving rise to gastrula, that is primary germ layers. How many primary germ layers are there? 3 primary germ layers. Outer part is called epiblast, mesoderm is middle and inner is called hypoblast. Understand? So either epiblast or ectoderm give rise to all these structures. Either mesoderm give rise to another and hypoblast or endoderm. Sometimes hypoblast they will ask or sometimes they will ask endoderm. In both they will ask means another option will be both A and B. Is that clear? So that is about this development of the inner cells. Clear? Now how long this will be developing inside? That I already told you. 280 days, 40 weeks. Okay. So here among that 40 weeks we are dividing into 3 areas, 3 types. One is called first trimester, second trimester and third trimester. Whole pregnancy, entire pregnancy is divided into 3 segments. Below same page, below last paragraph it is the 280 days gestation. 40 weeks. Gestation means the pregnancy period is called gestation period. Okay, now the first trimester, it is a main period of organogenesis. So this organogenesis takes place in the first trimester period. What is organogenesis? Formation of organs. What are all the organs? Heart is developed. Okay, same. If you find the heart, limb, lungs, liver, external genitalia, organ are all well developed. Okay, same page, first paragraph, last third line you see. Same page, first paragraph, last line. The embryonic heart developed during 4th week of pregnancy. Okay, 4th week of pregnancy means that means here, first trimester. Understand? Because there also heart is given, the above line also heart is given. Right. Now the second trimester, all the eye related to things are all developed here, eye. So eyelids, eyelashes, eye blinks, then also some hair growth will be there, then body is covered by fine hair, like muscle tissue, bone becomes stronger, all this. And the last trimester, here you will have fully developed baby. Okay, so when you study the germ layer, you study the types of pregnancy also together. Type of pregnancy, that how the 40 week is divided into 3 and each trimester what is the purpose of it. So these are all very important regarding this. So we studied the entire structure of the maintenance of pregnancy, that is how long the pregnancy is taking place, what are all the things happening, by the time simultaneously inner mass of the embryo, what are all the things are happening. Okay, now another last content in this is, we have not studied about this trophoblast. What the trophoblast is going to happen. We studied only about the inner mass of cells, because the entire blastocyst will have 2 things. One is the trophoblast, another one is called the inner mass of cell, 20 to 30 rounded cell. Now this upper part, trophoblast, will give rise to extra embryonic layer that we will talk now. Okay. Okay, now the last part is extra embryonic layer. Now here listen, we studied this full structure is called the blastocyst. Inner to the blastocyst we have the embryo. Embryo is formed into 3 germ layers: epiblast, mesoderm, hypoblast, and the entire pregnancy for 280 days, 40 weeks, 3 trimesters. Okay. Now this outer layer, it is called the trophoblast. That we know, trophoblast. The trophoblast, the inner layer gives an embryo. That means the entire fetus is formed from the inner layer. This fetus is protected by some layers outside the embryo. So it is called extra embryonic layers. So always trophoblast only forms this extra embryonic layer. At the same time, the trophoblast will protect the developing embryos. Okay. So now there are 4 types of extra embryonic layers are there. That means the trophoblast forms 4 different outer covering to protect the developing embryo. Okay. So extra embryonic layers are formed from trophoblast. What is the role of trophoblast? It protects the developing embryo by making a cover on it. Okay, above the embryo. And this cover, how many covers are it is made? 4 covers it is made. Okay, that we will talk now. Now this diagram represents this entire extra embryonic layers formed by the trophoblast. Now this part is fixed into the mother's uterus. Okay, I have just put it like this. You could do like, and this last part is fixing on the mother's uterus. Okay, and this embryo is here now. So now this end part and this embryo are connected by a structure called placenta. Okay, placenta is from mother's part which is connecting the fetus. Okay, right. So now here there are 4 structures one will study, then we will go for the placenta. The first, this is the embryo, which is the inner mass of cells, embryo. Okay. Now the first layer which is formed like this is called the allantosis. In your book it is ulta, it is given, but first layer is the allantosis. Okay. See, and the close by it is called yolk sac. The second layer. We call these 2 things together, it is called the umbilical cord. The umbilical cord means, the umbilical cord, these 2 things, that means allantosis and yolk sac together form umbilical cord. The umbilical cord is from the fetus side which is connecting to the placenta. Okay, umbilical cord will connect it to the placenta. At the time of delivery, the umbilical cord only is cut. Okay, so placenta is the mother's, where the endometrium is there, that part it is. Clear? And the placenta and the fetus is connected by, this is the fetus, this is the placenta. These 2 things are connected by the umbilical cord. Okay, that means the shape of the placenta is disc shaped, where you will find mother's blood also will be there. From mother's side, mother's blood will be there. From fetus side, fetal blood will be there. Understand? Umbilical cord from fetus side, placenta is from mother's side. So placenta is connecting to the disc shaped, umbilical cord is connected to the disc shape of the placenta, which has maternal means mother's blood as well as the fetus blood in this. Okay, right. Now the role of this embryo, why it is kept inside this cover is, the embryo will often affect by certain mechanical shock. When the mother is walking, when the mother is running, sleeping, that time it may lead to some shock. So to prevent that and also to absorb some nutrition from the mother's blood, exchange of gases, for all this it is being given a kind of a protection. Okay. So now when you talk about the allantosis, allantosis is small out pocket coming out. Okay. And it is the caudal end of, that means where the end of the yolk sac is there, from there it starts. Caudal end of the yolk sac. It is a small out pocket, it is present on the caudal end of the yolk sac, form the umbilical cord, join the placenta. That much clear? So if we can write what is allantosis when you write it. Second one is yolk sac here. The yolk sac will have earliest blood cells and blood vessels will be there. Blood tubes, blood cells will be there, early blood cells. Okay, right. And the third one is this layer which is filled with a fluid called amnion. A fluid which is surrounding the fetus called amnion. Okay. Now it is giving kind of a protection, at the same time it supplies some nutrition and all this. And the last outer part is called chorion. It is the outermost end of this trophoblast. Okay. And this outermost layer only will be connecting towards the it's a finger-like structure like this. See, this is a finger-like structure. This outer part only will be leading to this finger-like structure called chorionic villi, and where it is connected to this placenta. Okay, so outer part is a chorion where it is having a finger-like projection towards the placenta, and this projection is called chorionic villi, and it will be connected to the placenta where the exchange, that means maternal blood also will be there, fetus blood also will be there. Understand? So this is what the extra embryonic layers are. So extra embryonic layers are formed from the trophoblast, and there are 4 type of trophoblast are there. Layers are there. Allantosis, yolk sac, together it is called umbilical cord. Then amnion, fluid filled, where the embryo can float on this. It gives a kind of a buoyancy, that means a floating kind of a nature, because fluid is there. The embryo can float on it in order to avoid all this mechanical shock. So when the embryo, if it is water, this embryo is just having a shock towards the wall, it does not happen anything, it is not affected because of the fluid is filled. And the last one is called the chorion, where it is connected by a finger-like structure called chorionic villi. It is connected to the placenta. Clear? Right. So that is what we have concentrated today. We studied what is fertilization, we know what is implantation, maintenance of pregnancy, and in that maintenance of pregnancy, how many days pregnancy, what are all the 3 characteristics in it. Then we also discussed about the inner mass of cells as well as the trophoblast, extra embryonic layer. The extra embryonic layer separately they will ask you a 5-mark question. Fertilization, implantation, most of the time they will ask some 2-mark questions. What are all 2-mark questions are there? You know, capacitation is, what is fertilization? Then capacitation, agrosomal reaction is there. Then polyspermy, how you can avoid polyspermy. Then sometimes 5-mark question will be asked cleavages. Cleavage means zygote, blastomere, morula, blastocyst, and inner mass, all this. Okay. Then maintenance of pregnancy. Sometimes they will ask you regarding your 3 germ layers as well as the total pregnancy period. Right. The last part is placenta is acting as a last paragraph it is given, and first paragraph also, second, third line it will be there. Placenta will be acting as a temporary endocrine gland. See, the placenta is a connection between the umbilical cord. Okay, it is connecting the umbilical cord to the fetus. Umbilical cord connecting the fetus to the placenta, mother's fetus. Okay, right. Now here, the placenta is an endocrine gland, that means it can produce some hormones at the time of pregnancy. That time of pregnancy only it synthesizes, produces an hormone, because placenta developed only at the time of pregnancy. So it produces an hormone called, separately 2-mark question, book back question, it is human chorionic gonadal tropic hormone, HCG, expansion of right. Okay, and second hormone is human chorionic somatotropin. Okay, then human prolactin, this gonadotropin hormone. Okay, so that is also important. All these 3, apart from this, that is HPL, that is prolactin, lactogen hormone, the last 3rd one. Apart from that, you will have certain hormone called estrogen. Then you will have progesterone. All these are during your time of pregnancy time. Then one very important called relaxin. This hormone is produced or secreted at the time of delivery, where it will relax the smooth muscle of the pelvic area, pelvic ligament. So these are all the hormones which is produced by the placenta. Book back question. Okay, when they ask this book back question, you have to write first paragraph, 3rd line, then last paragraph fully. Third, you will find some hormones also, extra hormones also will be given in the end of the paragraph. Okay, so that's all today, and tomorrow's class we will be discussing about the male reproductive organ, then we will go for the female reproductive organ. Within 2 classes we will complete this entire human reproductive system. Okay. Now to take your note for your recap. So far what we studied, the same. So far what we studied, we will have the same. Question number 1. Question number 1. Polyspermy is avoided by dash membrane. Polyspermy is avoided by dash membrane. Polyspermy is avoided by dash membrane. Question number 2. How many cells form the embryo or the inner mass of the cell? The inner mass of the cell contains how many cells? Inner mass of the embryo contains how many cells? Question number 2. Okay. Question number 3. The pregnancy that takes place outside the uterus are called dash. The pregnancy that takes place outside the uterus, not in, outside. The pregnancy that caused outside the uterus. Okay. 4th question. Dash extracellular or extraembryonic layer giving rise to early blood cells and blood vessels. The dash extracellular layer giving rise to early blood cell and blood vessels. Which cell? Which layer? Last question. Last question. Last question. Mammillary gland is produced by dash germ layers. Mammillary gland. Mammillary gland is produced from dash germ layers. Mammillary gland is produced from dash germ layers. Check the answer. Polyspermy is avoided by fertilized membrane. Question number 2. Inner mass of embryo 20 to 30 rounded cells. 20 to 30. 20 to 30 rounded cells. The pregnancy, 3rd question. The pregnancy which takes place outside the uterus are called ectopic pregnancy. The earliest blood cells and blood vessels are formed from dash extra embryonic layer, that is the yolk sac. Yolk sac only giving rise to early blood cells as well as blood vessels. And the last question is, the mammillary glands are produced or synthesized by a germ layer called ectoderms, or otherwise called as epiblast. Epiblast will be giving rise to it. Clear? Right. Thank you.